majority of Chapter 4 today. There's going to be a part kind of in the middle of Chapter 4 that I'm going to skip over, gloss over, and we'll come back to look at that next time. And then for next time, we're going to be talking about a case study, an Ebola case study, so don't let me forget to give that out at the end of class today. So get that and take it with you so you can read that over before class on Thursday. Assuming we have class on Thursday, there's more of the white stuff coming on Wednesday. So, Chapter 4, we are talking about replication. So, we went through Chapter 2, where we talked about all the protein capsids. We've gone through Chapter 3, we talked about all the different genomes. So now we've kind of put our two pieces of our virus together, and now in Chapter 4 we're ready to start talking about the replication strategies. So we will talk about, there are 7 stages that every virus goes through as it replicates. We're going to go through those 7 strategies today, hopefully all today, and then we will use Ebola as a case, as a example when we talk about the case study on Thursday, and we'll kind of walk through these 7 strategies specifically for Ebola. So this little diagram shows you a good chunk of these strategies. So we know that the first step or the first phase is going to be attachment, where the virus interacts with the cell. The viral genome has to enter the cell, so that's what we see here. Viral replication has to take place, so the genome is replicated. Proteins are translated, so that's what we have here. Viral mRNA is translated, proteins are processed, and we have assembly and maturation release of mature virus particles. That's sort of a very generic overview of virus replication. So here are our learning objectives for Chapter 4. We want to understand how the nature of the virus genome determines its pattern of replication. So that's the part that we'll kind of save for next time, where we talk specifically about the different classes of viral genomes and how that contributes, how the way that they are categorized specifically contributes to the replication strategy. Today we'll kind of walk through a typical replication cycle, and then we'll be comparing the patterns of those 7 major classes next time. Okay, so remember we talked about way back on the first day when we were in Chapter 1, we said that the sole objective of a virus is to replicate. Its main goal is to get inside a host cell and replicate the genome so that it can produce more infectious virus particles that can spread throughout its host. So that is the crux of Chapter 4, is we are talking about how do viruses do this. This is their sole objective. There are 3 things that influence how a virus replicates. Number 1 is the type of cell that is infected. So is it a eukaryotic cell? Is it a prokaryotic cell? Is it an animal cell? Is it a plant cell? Is it a kidney cell? Is it a digestive tract cell? So what type of cell becomes infected? The second thing that influences viral replication is the capacity of the genome. So remember in Chapter 3, we talked about the fact that viruses range pretty dramatically in the size of their genomes. There are some viruses that pretty much bring everything they need with them into the cell in order to be able to replicate. So these are your large viruses, your adenovirus, your herpes virus. These viruses pretty much bring into that cell with them everything that they're going to need. Then you have your smaller viruses that have very tiny genomes that are much more reliant or dependent on their host cell in order to carry out their replication strategy. So the capacity of that genome can play a major role in how that virus replicates and how it interacts with the host cell. And then lastly, the type of genome. Is it a DNA virus? Is it an RNA virus? Is it single-stranded? Is it double-stranded? Is it positive sense? Is it negative sense? All of those things are going to play into not only how that virus replicates, but where within the cell that virus replicates. So for example, if I'm a DNA virus, I have to make my way to the nucleus once I get inside a host cell. If I'm an RNA virus, I don't have to go through that extra barrier or that extra step. So the type of genome is going to play a role in how viruses replicate. Now, the way that we study viral replication strategies, we mostly use bacteriophages. They are a really good model for us to use to study how viruses replicate. So most of the work that has been done looking at this and developing these strategies and looking at specific viruses and determining how they replicate has been done using bacteriophages infecting bacteria. It's a simple system, right? Our host cell is a simple prokaryotic bacterial cell, so there's not a lot of complexities in terms of how the virus interacts with the host. So it's a simple model system that we can use to study how viruses infect bacteria. So that has led us to these three sort of essential phases of replication, and we're going to flesh these out a bit as we go through the chapter today. But the overall three phases are initiation of infection, replication and expression of the virus genome, and release of the mature variants from the infected cell, so into the host. So those are sort of three major overall phases, and then we're going to flesh those out as we look at the different strategies. So here are our 8 stages of replication. So we have attachment, penetration, uncoating, genome replication, gene expression, assembly, maturation, and release. So often gene replication and gene expression are sort of combined into one strategy. I separated those out because we're going to talk about them separately as we go through today. So this being the cycle, we could pretty much start anywhere, but it kind of makes sense to start at attachment. So again, this figure is from your book, kind of walks you through these 8 phases. Attachment is what you think it is. This is where the virus interacts with or attaches to a specific binding, through a specific binding point to the cellular receptors. Remember we talked about there being a VAP on the capsid, on the outer surface of the capsid, so that's the virus attachment protein, and that virus attachment protein is going to interact with a specific cellular receptor on the host cell. So this is what we coin attachment. The target receptor molecules are typically glycoproteins. So these are the receptors on the host cell are typically glycoproteins, or the carbohydrate residues present on the glycoproteins. Okay, so this is on the host cell. When we're talking about the host cell has membrane or has proteins on the membrane which serve as cellular receptors, and it is these receptors that the virus targets or manipulates in order to achieve attachment or interaction with that host cell. So it's typically the glycoproteins themselves or the carbohydrate residues present on those glycoproteins. So that can kind of confer some specificity for the virus. If I'm a virus and I target a specific glycoprotein on a specific cell, then that is a much more specific interaction than if I am simply targeting the carbohydrate residue on a glycoprotein, right? There may be more than one protein on this cell that has the same carbohydrate residue. Okay, so here I've got 3 different proteins. They all have the same carbohydrate residue on the tip of them, but they're 3 different proteins, right? So if I'm only caring about that carbohydrate residue, I have less specificity of the specific cellular receptor that I'm interacting with. And this can be a good thing for a virus, or it could be a bad thing for a virus. It depends. And we'll talk more about that later on. Also, there are some viruses, some complex viruses, that have more than one receptor or have alternate routes for uptake inside a cell. So this is just kind of another layer of flexibility where these viruses have even less specificity, and where they, or sometimes they require two interactions in order to completely interact with and penetrate that cell. So again, these are going to be your more complex viruses, herpes virus, pox viruses, for example, require more than one receptor in order to interact with the cell. The movie that we watched on Friday on HIV, remember there were two receptors that HIV needed to interact with in order to get inside that cell. So again, that sort of adds a layer of specificity for those viruses to make sure that they are interacting with a true potential host cell. All right, so let's talk about some of these receptors that are present on the host cell that interact with the vat. So there are many different types of receptors that can serve as a potential interaction point for a virus into a host cell. So lots of different classes, lots of different variations in these receptors, as you can see just in the simple structures here. The only thing that all of these receptors have in common is that they are not manufactured by the cell, they did not evolve, they are not there to interact with viruses. That is not why your cell puts them there. Okay, they all have other jobs that they are supposed to do, other things that they are supposed to interact with in order to help that cell survive, or they all have other jobs that they do within that cell. So that is the only unifying feature of these receptors, is that they are manipulated, they are subverted, they are overtaken by VAPs, coerced by viruses to allow the virus to enter the cell. So they're all manipulated by viruses. The arrows here sort of indicate where the virus attaches to these different receptors. So number 5 is a big one that we will talk about, that is integrins. You might have heard of integrins in your cell bio course. So number 5 is, we'll talk more about that one later on. Number 8 is also a big one that we'll talk about in just a second. That is sialic acid. Anybody know what virus interacts with sialic acid? Influenza. So this is the receptor that influenza uses in order to interact with host cells. We'll talk more about that next. A lot of these a lot of these receptors, by the way, these first 4, for example, again, another thing that you notice about many of these viruses is they are transmembrane, meaning they span the membrane. They have both exterior regions of the receptor and interior regions. So a lot of them are transmembrane receptors. A lot of them are involved in adhesion, so that's like the integrins, cell adhesion molecules. These are all really good receptors that viruses use to allow for attachment. So I'm going to talk a little bit more about that sialic acid attachment with influenza. So again, this is so this little blue. So these little blue protrusions here, these are the sialic acid receptors on the surface of the host cell. And then this big red blob right here, that's your influenza virus. So if we zoom in on that influenza virus a little bit, you can see we have these little spikes on the outer surface of that virus. So that is what these things are. So those spikes are formed by a hemagglutinin glycoprotein. So it is present in a trimer. So 3 hemagglutinins come together to form the VAP for influenza. So hemagglutinin is the VAP for influenza. This is how influenza interacts with the sialic acid receptor on the host cell. Now the problem with this is that a lot of cells have sialic acid receptors on the surface. So there is very little specificity inferred here by this VAP receptor interaction. So influenza can infect lots of different cells, a very wide range of cells when it gets inside your body, right? Who's had the flu before? sort of symptoms did you have? Nobody was sick. Coughing, fever, throwing up, sweating, headache.
I had pneumonia.
Sick stuff, right? So have you ever met somebody who you had the flu and they had the flu and your symptoms were completely different? Yes, right? So the impact of influenza on different individuals can be completely different. Now that's due to a couple of different things, which we'll talk about more when we get to chapter 6. But one of the reasons that happens is because influenza can infiltrate almost any cell in your body. It can attack the gut, it can attack the respiratory system. There are lots of different places that influenza can find potential host cells in your body because of the way that it attaches. Because of this sialic acid receptor that it interacts with, which is present on lots of different cells inside your body. So its host cell pool is varied and tremendous once it gets inside. Now, influenza is one of these viruses that actually requires sort of a two-pronged approach to attach and completely penetrate your host cells. So hemagglutinin interacting with the sialic acid receptor, that is the first prong of this approach. And the other glycoprotein on the surface of the influenza virus is called neuraminidase. And neuraminidase is sort of the second prong of this approach. It is required for the next phase, which is actually penetration. And so in order for the virus to actually penetrate the host cell, neuraminidase also has to come down and interact with the sialic acid receptor. Now, there are times when influenza interacts with a host cell through the sialic acid receptor, but then realizes that this host cell is not going to be a good host for it. Okay. What would a virus require in a good host cell? It's kind of a very broad question.
Yes.
What situation could you think of that influenza would bind to a cell that had the sialic acid receptor and then decide not to enter that cell? Okay, so if it is an old cell, and by old we might mean it's not metabolically active, right? So it could be that it is a cell that is dying, that it is a cell that is not dividing, not metabolically active. It could be that it's a cell that is kind of dormant and not actively replicating, right? In order for a virus to reproduce, we need a host cell that is metabolically active, that has all the cellular machinery that we need, that is going to be carrying out replication and transcription and translation. So we need a host cell that is biochemically and metabolically active. Not all cells are. So neuraminidase is sort of the exit strategy for influenza. Once it binds to a host cell, it can determine whether that host cell is metabolically active, and if it's not, neuraminidase cleaves the sialic acid and allows influenza to detach from that host cell and not enter the host cell. So it's sort of the exit strategy for influenza so that it can go on and infect another cell. So it's kind of have that two-pronged approach, pretty sophisticated approach for a virus like influenza to be able to choose a potential host cell. Now, rhinovirus is another really interesting virus when it comes to attachment. Anybody know what rhinovirus is responsible for? The common cold, right? The sniffles. The common cold. So this is the rhinovirus. Rhinovirus has a really interesting conformation where the proteins of the capsid come together to sort of form this V-shaped structure and this canyon-shaped structure. So the receptor here, so this is on the host cell, it's called ICAM. So this is a cell adhesion molecule, kind of typical of those cellular receptors that we see. So this is on the host. This red part would be indicative of being on the host cell. The blue part here, this is all virus. So VP1, VP2, and VP3, those are viral proteins. Those come together in such a way that they form this V-shaped structure or this canyon. And the interaction point for that cellular receptor is right here in the floor of that canyon, okay, in the depth, far depth of that V. Now this is really interesting for rhinovirus, and it's actually a really good feature for viruses. So what does your body use to neutralize viruses? If we get a viral infection, what's sort of our first line of defense? Antibodies, right? So if I get rhinovirus, the first thing that my immune cells are going to do is start producing antibodies against that virus. What do you know about antibodies? Okay, they produce when they send something that's foreign, but you have to have some history of it. Nathan, you said they're small. You guys agree with that? Megan, what were you going to say?
They need like space to like bind.
They need space to bind. In fact, antibodies are huge proteins, right? They're big, blobby proteins. Most of the time, remember, we draw them. They have that Y shape, right? Because we've got the heavy chain, the light chains, the IgGs. Antibodies are relatively big proteins. So why do you think this sort of conformation for rhinovirus is going to be advantageous for it? The antibody can't get in there, right? Do viruses want to bind antibodies? No, right? If I'm a virus, I want to be able to avoid interacting with your antibody, because that antibody binds with me, it's going to neutralize me, right? Or I'm not going to be effective. Some people say kill, but then get all upset with the virus people who don't think viruses are alive. Like, how can we kill something that's not alive? So we use the word neutralize to make everybody happy. Okay? So the antibody binds to that virus, it's going to be neutralized. Okay? So I don't want an attachment site where an antibody can get in there and bind. Right? So this is really advantageous for me, because the only thing that can fit inside this binding pocket is ICAM, right? Is my receptor that I want to bind to. Nothing else can get inside there. What else do you know about viruses and particularly how viruses avoid being neutralized? What's one way that viruses avoid neutralization by the immune system? What do they do? Okay, so their genome often mimics host cell or cellular genome, so they often are kind of incognito, in disguise. What else do viruses do to avoid detection and neutralization once they get inside you? Yes.
Do they hide in the nervous system?
Some of them hide out in the nervous system. Yep. Herpes virus is classic for doing that. What else do they do? Think about HIV. What did we talk about HIV does? Mutate, right? They mutate all the time. Right? How many of you got the flu shot this year? The flu vaccine? Anybody still get the flu? My daughter did. The last 2 years, she has gotten the flu shot and gotten the flu. So she's like not on the vaccine board anymore. But it happens, right? So a couple of reasons why that happens. One of them happens to be that the virus can mutate, right? So the virus that we put in the vaccine is not the virus that is actually spreading around, because it has mutated, it has changed. HIV is notorious for that, because it's a retrovirus. We know that's a highly error-prone process. So as soon as we get good drugs that we're like, oh yeah, this cocktail is gonna work really well, it's gonna bind here, it's gonna stop that, it's gonna interfere with that, and next thing we know, HIV is mutated in such a way that our drugs are no longer effective. Now, so viruses are constantly mutating their antigens, right? So what does the antigen do again? Binds to the antibody. So it makes sense, if I'm a virus, I'm gonna constantly be changing my antigen so that your antibody can't bind to me, right? But if I'm a virus, do I want to constantly change my VAP?
No.
No, right? I don't want to mutate my VAP because then I won't be able to do what?
Infiltrate.
Interact with my host cell, right? I can't let my VAP get mutated because now I can't interact with ICAM. So this, I have to keep the same. I got to keep this same conformation. I got to keep those proteins the same so that I can interact with ICAM. But my antigen, I'll change that all day long. Like I'll mutate that all day long so your antibody can't bind to me. So what if I am a virus? This is virus, and what if this is my VAP and my antigen is right here? What problems do you think that might present for me? That's exactly right. I run the risk of when I mutate the antigen, I could mess up with that, right? So let's say I mutate my antigen in such a way that it now looks like that. What have I done?
You basically blocked out
I've blocked my VAP, right? So now my big blobby cellular receptor can't get in here, right? It's blocked. Okay, so if I'm a virus, I want my VAP as far away from my antigen as possible. Here's my antigen, here's my VAP. Now I can mutate this guy all day long without running the risk of interfering with my VAP. All right, and Rhinovirus is a really good, does that really well. It's totally separated its VAP from an antigen. This VAP is the design or the structure of that VAP is really specific. Only ICAM can fit in there. No antibodies can get in there. And its antigen is far away so that we can mutate that antigen all day long and not affect the VAP. Right? That's one of the reasons why there's no cure for the common cold, right? Like we don't have antiviral drugs to treat rhinovirus. We don't have a cure for rhinovirus. What do we do when we get infected with rhinovirus?
Cough medicine, cough syrup, try to
We
deal
take
with
medication to treat what?
Symptoms.
The symptoms, right? It does nothing to get rid of the virus, but we take medication to treat our symptoms. That's why you don't go to the doctor when you get a cold, because there's nothing they can do. Alright. Adenovirus is another interesting one. I'll zoom through that one. It also uses a two-pronged approach. So it has glycoproteins on the surface of the virus. So that would look like this. Is your glycoprotein. The CAR is the Coxsackie-virus adenovirus receptor. So that is the receptor on the host cell. So the CAR is on the host. So it interacts initially with the glycoprotein on the adenovirus. And then this is the structure, this icosahedral structure of the adenovirus interacts with integrins. So these yellow things are the integrins which interacts with actually the base of that virus. So you have to have both of these interactions occurring in order for adenovirus to enter the host cell. So this is an example of where you have to have, again, sort of this two-pronged approach. You have to have both the glycoprotein interacting with the CAR, and then the base of that icosahedron interacting with the integrins in order for adenovirus to enter the host cell. So again, just another level or layer of specificity for adenovirus. Questions so far? Okay, let's keep going. Alright, cell tropism. So this is basically, what we mean by tropism is the type of host cell that a virus can infect. So the expression of receptors on the surface of the cell determines the tropism of a virus. For example, the type of host in which that virus is able to replicate. So for example, if I'm an adenovirus, I cannot infect a cell that does not have both that CAR receptor and integrins. Right? I've got to have both of those things in order to find a potential host cell. If I'm influenza, I have to find a host cell that has the sialic acid receptor on it. Otherwise, that is not a potential host cell for me. Okay, so the type of receptors that are present determines whether or not I can replicate within that cell. So the attachment is, a lot of viruses argue, or a lot of virologists argue, is the most important phase within this replication cycle, right? So it's hard to judge, right? There are lots of different stages and every one is essential. But a lot of people argue that attachment is the most important because it has major influences or is a major determining factor on the entire course of that virus. So is it going to be an acute virus like the Rhinovirus that is gone within a couple of days, or is it going to be a virus like herpes virus that is with us for the rest of our life? A lot of virologists say all of that is determined by attachment, right? And this is the most important phase. A lot of antiviral drug target the attachment phase, right? Because if we can prevent the virus from getting in, we don't have to worry about any of the rest of these phases, right? If we can block this virus at attachment, we don't have to worry about blocking replication or assembly or any of those things. So a lot of antiretroviral targets actually target attachment. Tamiflu, right, that you get prescribed when you have influenza. That is a neuraminidase blocker. So it blocks the attachment phase of influenza and prevents influenza from entering the cells. That's why you can only take Tamiflu when? What do you have to do in order to get Tamiflu? You've got to go to the doctor early, right? Like as soon as you start to feel symptoms, if it's beyond 48, 72 hours, your doctor is not going to give you Tamiflu. Why?
It already.
The virus is already in, right? Tamiflu is not going to do anything for you at that point. It's already in your host cells. It's already replicating. It's not going to help you. All right, if you get Tamiflu within the first 24 hours, you can block a lot of those virus particles from ever entering your host cell, and you can shorten your duration or lessen your symptoms. But if you wait till you've had the flu for a week, Tamiflu is not doing you any good because all the virus particles are already in their host cells. Okay, let's talk about penetration. So penetration is the second phase. Penetration and attachment often blur together. So they occur, penetration almost immediately follows attachment. It's not like a virus is just gonna, bind to my cellular receptor and just kind of like hang out there for a few days, right? As soon as that virus attaches to a cellular receptor, it is going to penetrate the host cell. So penetration is actually a little bit difficult stage to study because it so closely follows attachment. We do have three main mechanisms for penetration. Now the difference between penetration and attachment, one big difference, is penetration requires that the cell is metabolically active. So if I'm a virus, I can attach all day long to any cell that I come in contact with, but I cannot penetrate that cell unless it is metabolically active. right? So, that's one big main difference. There are three main mechanisms that we have, ways in which viruses penetrate the cells. So the first one is simple translocation. This is where the entire virus particle is simply flipped or translocated across that membrane. So it interacts with so here's our receptor in blue. Here's our virus in red. So the virus is going to interact with the cellular receptor. It binds to the cellular receptor, and then the entire cellular receptor and virus is flipped inside the membrane. The virus is released to go off into its thing, and the receptor is recycled and put back on the exterior surface of the membrane. So this process is mediated by both viral proteins and cellular specific membrane receptors. So it requires both virus proteins and host cell receptors working together to carry out this process. This is a relatively rare strategy. We don't know if a lot of viruses actually use this strategy. We don't really have a good understanding of how this works and how it happens. Again, penetration is kind of a hard strategy to study because it happens almost simultaneously with attachment. The other two strategies that we have are actually much more common, and we know a lot more about them. So endocytosis, you guys have all talked about endocytosis before. We know that it is dependent upon clathrin-coated vesicles that travel close to the cell membrane. We have our virus particle here that's sort of infiltrating that membrane, kind of causing this indention in the membrane, and this part of the membrane sort of pinches off, and now we have our virus inside the clathrin-coated vesicle on the interior of the membrane. Again, we know that this requires ATP, so this requires that cell be metabolically active. You couldn't do this in a cell that is not metabolically active. Now, if a virus is enveloped, right, so if it already has a lipid membrane surrounding it, fusion has to follow endocytosis, right? So if a virus is not enveloped, right, if this is just genome and capsid, how does the virus get out of that clathrin-coated vesicle?
By budding?
Not by budding. How do things get out of vesicles once they get inside host cells? Megan? The vesicles get lysed, right? One way or the other, the vesicle has to lyse in order to get that virus out, okay? So oftentimes these vesicles will fuse with the endosome. Sometimes they will fuse with the lysosome, depending on what's going on. And whatever is trapped inside this vesicle will be able to get out. Now, so that's the way that the virus would do it as well. Now, if that virus has a lipid envelope already, if it is an enveloped virus that already now has a lipid membrane surrounding it, now inside